Short questions and answers, nucleic acids and related topics. Nucleic acid. 1. Who discovered nucleic acids and when? A Swiss physician discovered nucleic acids in 1869. 2. Where were nucleic acids first isolated? From the nuclei of pus cells, white blood cells. 3. What was the initial name given to nucleic acid, and why? Nuclein, because it was found in the nucleus. 4. Why was nuclein renamed nucleic acid? Due to its acidic nature. Chemical structure of nucleic acids. 5. What are nucleic acids? They are linear, unbranched polymers made of nucleotides. 6. What are the two types of nucleic acids? DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. 7. What are the monomers of nucleic acids called? Nucleotides. 8. What is the composition of a nucleotide? A pentose sugar, a phosphate group, and a nitrogenous base. 9. What is the sugar found in DNA? Deoxyribose. 10. What is the sugar found in RNA? Ribose. 11. What provides the acidic property to DNA and RNA? Phosphoric acid. 12. What are nitrogenous bases? Ring structures containing nitrogen that can acquire a proton due to unshared electron pairs. 13. What is a nucleoside? A molecule formed when a nitrogenous base is linked to the 1' carbon of a pentose sugar. 14. What is the difference between nucleoside and nucleotide? A nucleoside lacks the phosphate group, while a nucleotide includes it. 15. What are nucleoside monophosphates, diphosphates, and triphosphates? Nucleotides with 1, 2, or 3 phosphate groups, respectively. Nitrogenous bases in nucleotides. 16. What are the two classes of nitrogenous bases? Pyrimidines and purines. 17. Name the pyrimidine bases. Cytosine, C, thymine, T, and uracil, U. 18. Name the purine bases. Adenine, A, and guanine, G. 19. Which nitrogenous base is found only in DNA? Thymine. 20. Which nitrogenous base is found only in RNA? Uracil. 21. What happens to the two terminal phosphates of a nucleotide triphosphate during DNA or RNA polymerization? They are lost during incorporation into the nucleic acid. Phosphodiester bond formation. 22. What type of reaction joins nucleotides together? Condensation reaction. 23. What is released during nucleotide polymerization? Pyrophosphate, two phosphate groups bound together. 24. What drives the polymerization reaction of nucleotides? Hydrolysis of pyrophosphate, releasing energy. 25. What is a phosphodiester bond? A bond between the phosphate group of one nucleotide and the 3' hydroxyl group of another. 26. What are the ends of a polynucleotide chain called? The 5' phosphate end and the 3' hydroxyl end. ATP and NAD. 27. What is ATP? Adenosine triphosphate, the energy currency of the cell. 28. What are the components of ATP? Adenine, base, ribose, sugar, and 3 phosphate groups. 29. What happens during ATP hydrolysis? ATP is converted to ADP and inorganic phosphate, Pi, releasing energy. 30. What is NAD? Nicotinamide adenine dinucleotide, a coenzyme. 31. What is the composition of NAD? Two nucleotides joined by their phosphate. Watson and Crick model of DNA. 32. What is Chargaff's rule? The amount of adenine equals thymine, and the amount of cytosine equals guanine. 33. What is the width of the DNA double helix? 2 nanometers. 34. How many base pairs are in one turn of the DNA helix? 10 base pairs. 35. What type of bond holds base pairs together? Hydrogen bonds. 36. How are the DNA strands oriented? Antiparallel, with one strand running 5 end to 3 end and the other 3 end to 5 end. 37. What is the base pairing rule in DNA? Adenine pairs with thymine, and guanine pairs with cytosine. 2.26 ribonucleic acid, RNA. 38. How does RNA differ from DNA? RNA is single-stranded, contains ribose, and has uracil instead of thymine. 39. What are the three main types of RNA? Messenger RNA, ribosomal RNA, and transfer RNAL. Roles and types of RNA. 40. What is the function of messenger RNA? Carries genetic information from DNA to ribosomes for protein synthesis. 41. What is the function of ribosomal RNA? Forms part of the ribosome and provides a platform for protein synthesis. 42. What is the structure of transfer RNA? A single-stranded molecule with a cloverleaf shape. 43. What is an anticodon? A sequence of 3 bases in transfer RNA complementary to a codon in messenger RNA. 44. How many different transfer RNAs are found in human cells? About 45 different. Central dogma of life. 45. What is the central dogma of life? The flow of information from DNA to RNA to protein. 46. What is gene expression? The process by which information from a gene is used to synthesize proteins. 47. What are the two main steps of gene expression? Transcription and translation. 48. What happens during transcription? mRNA is synthesized from a DNA template. 49. What happens during translation? The mRNA sequence is used to synthesize a protein. 50. What is a codon? A sequence of 3 nucleotides in mRNA that specifies an amino acid. Conjugated molecules. 51. What are conjugated molecules? Molecules joined with other types of molecules. 52. What are glycolipids? Lipids attached to sugars, found in the brain and myelin sheath. 53. What are glycoproteins? Proteins attached to carbohydrates, functioning as hormones, transport proteins, and receptors. 54. What are lipoproteins? Complexes of proteins and phospholipids, found in milk, blood, and egg yolk. 55. What are nucleoproteins? Complexes of proteins and nucleic acids, found in chromosomes and ribosomes.